It's a pleasure and an honor for me to be here and introduce you the talk of Dr. Cunningham. So I had the pleasure to work during my PhD with Dr. Cunningham around 9 months and learn a lot about Salmonella biology and infection. And it was actually one of the milestones of my career, so I'm really happy to meeting him here again in Mexico after so many years. So as a brief introduction, Dr. Adam Cunningham is a professor of functional immunity at the University of Birmingham, a director of Back-to-Back, a global network accelerating bacterial vaccine development. His research defines how adaptive immune responses are initiated, regulated, and sustained following infection or vaccination, with an emphasis on antibody responses to bacterial pathogens, especially in Salmonella. He was trained in pathobiology and immunology at the University of Southampton, and he established his independent group in Birmingham in 2005 and was appointed professor in 2011. His work informs the rational design and evaluation of next-generation vaccines to address antimicrobial resistance, and he will talk to us today about how the inflammatory responses induced to Salmonella drive thrombosis. So welcome, Adam, and it's a pleasure to have you here.
Thank you very much, and thank you very much to Constantina and Christina for that very kind words. And it's absolutely wonderful to be here in this really, really beautiful environment and to share some of our work with you. And before we do, I'm just going to plug the network that was mentioned about Back-to-Back, which is a network which promotes the development of bacterial vaccines. And although I'm talking about thrombosis here, it's really important to remember that if we have decent working vaccines, what I'm going to talk about next isn't a problem. Okay, so this slide is just to get my thanks in early. These are a group of people who've worked on this project and collaborated on this project over many years now, and I'm exceptionally grateful to all their efforts. I'd just like to point out Marisol and Noni, who both came from the excellent research pipeline, which is within Mexico. Okay. So bacterial infections remain a leading cause of death. One study published a couple of years ago now showed that estimated that about nearly 8 million deaths globally were caused by just 33 bacterial pathogens. This equated to about 1 in 7 of all deaths globally. And of course, our ability to do anything about this is becoming more and more restricted, both in terms of the treatments that we have available, but also where we can use those treatments globally, access and so on and so forth. And when we think about, why should we die from a bacterial infection? Why should we suffer? Why should we experience any symptoms? Sometimes it's really to understand it. If we take the causative agent of tetanus here, it produces an exotoxin, and that neurotoxin has a specific known target. You take away the toxin, you take away the problem in the main. For other organisms, like the gram-negative organism Salmonella typhimurium, it doesn't have an exotoxin. So it doesn't have that simple pathway to really drive its effects on us. It does have endotoxin, most often referred to as lipopolysaccharide. And if you look where you find LPS, it's in the gram-negative cell wall. And if we take a step back and ask what evil function that LPS performs, what does it do to us that so cruelly causes such harm to us, then all it does is make the cell wall stable. And so it's actually the way that we see LPS, how we respond to LPS, that dictates the harm that it causes to us, and not an intrinsic toxin activity of that molecule at all. And that's really important, because when we look at infection, we can think about things in terms of outcomes, always killing the organism, but what does it do to us? So we've got 3 scenarios here. Here's our bug, and here it's in the environment of you or me, and each time. If we have a vaccine, then the vaccine acts like a sniper, and it takes out the pathogen, but it leaves the rest of us intact. We're not harmed by this, and that's why we don't know when a vaccine is working in the main, because it's working silently in the background. Under normal circumstances, when we respond to an infection, we either bring out innate or adaptive immune responses, and these kill the bug, but also do a limited amount of harm to you as well. And finally, we can go nuclear on this. Now, the key point about all these three scenarios is that you're taking out the pathogen in each one of them. The immune system is fulfilling its role of killing the target pathogen. But the harm it does to you, these processes do to you on the way, they vary quite significantly. And so cross-cutting all of this is, of course, inflammation. And when we think about this, we often think about it in the sense of immune cells acting, or we think these are good, and then we also think about mediators, and these are cytokines, and here are a couple of ones I'll be talking about, interferon gamma and TNF, and then we also have other molecules like IgG. But actually, these don't work in isolation in the main. They work in a functional, organized manner. And this is incredibly important because the whole immune system is set up to be organized. And so we can see this here, whether we're thinking about it at a gross level in terms of, as we have 700 or so lymph nodes distributed throughout the body to prevent antigen dissemination, as you can see here. Or you can have other presentations of this, like bacteria being brought together in the form of an immune complex, whether the bacteria are intracellular, either in or outside of a vacuole. Or if we now start going up in scale a little bit more, we've got granulomas, or an area that I'll be mainly talking about here is the concept is if we encounter pathogens in the vasculature and that we try and trap these within thrombus. So all of these are not necessarily about instantly killing the organism, but they are about mounting controlled immune responses in selected regions to prevent bacterial dissemination or control bacterial dissemination. And if we think about it, even if we just take a very simplified view of this, inside our gut, we have roughly a kilo or so of bacteria, but we don't necessarily care about mounting fulminant responses against those organisms because we have they are not affecting us systemically in that sort of simplified manner. Okay, so what I'm going to talk about today is the concept that all these encounters lead to impacts on the cardiovascular system. And probably if there is a couple of messages to really take home from this is this is a really important statement from Steve, who I showed you the picture earlier, is that nothing kills you faster than a platelet. Literally, you can be dead inside a few seconds from the consequences of these becoming activated. And that's important. So if you start joining a few dots together of what I introduced in the last couple of slides in this, is that we might think we don't have to kill every organism that we ever encounter. Our gut bacteria show us that that's the case. But we do need to control where they are. And this both buys you time to mount adequate immune responses, but it also means is that other activities like the cardiovascular system can go on and do its job, which is to keep you. So our immune systems in this context are acting both as a police officer to police what's going on inside normal physiological activities like the cardiovascular system, but also as the army to protect you from external threats. Okay. So we know that pathogens and products interact with the cardiovascular system. And one of the concepts of this is around this immunothrombosis, which is generally seen as being positive. Pathogens interact with various components of the immune system, resulting in platelet activation. You get pathogen trapping in that sort of classical model I showed you in a simplified cartoon earlier. But then you can also have thromboinflammation. And here, what you're really looking at is more of a dysregulated inflammatory response, where you get pathological clot formation and essentially cascading of inflammatory pathways. And so the key point here is, again, this underlying principle of control, and hopefully that will cross-cut everything I'm going to show about today. And I've mentioned this. So the model I'm going to talk about today is Salmonella. Most of the work is going to be Salmonella in a mouse, but there will also be Salmonella work using human material, and then one slide on COVID to sort of show how these studies bridge. So if you have a Salmonella gut infection and it stays there, you've got an incredibly low risk of death. If that bug, the same bug, just goes through the barrier and spreads throughout the host, and the bacteremia here is one organism per mil of blood, it's tiny. And that's enough to change your risk of death for typhoid to untreated typhoid is about 1 to 10%. For untreated invasive non-typhoidal Salmonella infections, it's about 1 in 4. These are staggering numbers when you think about it. And yet it's the same organism, and only its locational distribution is altered. And so there's a rich history of Salmonella infections being associated with cardiovascular and thrombotic complications. The oldest paper I've been able to find is about 120 years ago, old or so. But there's a whole range of these, but it's not really thought of as a byproduct of having a Salmonella infection. It's just the presentation and how it appears. But actually, even if we focus in on non-typhoidal Salmonella infections, those ones that stay in the gut, then study suggests is that you're still left with a significantly increased risk of one or more of DVT, pulmonary embolism, and stroke years afterwards. Okay. So on top of these can have multiple presentations. I'm largely going to focus on venous thrombosis, but these can result in organ failure and damage. And remember, even in that worst case scenario I gave you about invasive NTF, 75% of those individuals are likely to survive in that instance. Now, that means is that there's the potential for long-term damage to be inflicted on the host. And the impact of Salmonella infections in the mouse and in the human are really quick, at least on platelet numbers. You can see here, so mice have more platelets than humans, about 3 or 4-fold more. And then, but within a day, you can see is that there's a rough halving of the numbers of platelets, reaches its nadir between this day 7 and 21 period before it recovers in this recoverable model. And then you can see a reciprocal change in mean platelet volume. So the impacts of Salmonella infections, like you'll see in very many serious infections, is rapid. And because this stuff was published a few years ago, I just want to focus on what we originally found. And we found is that given infection drives a thrombosis in the liver that we detect at day 7 after infection. We don't see it at day 6, we don't see it at day 5, but we do see it at day 7, okay? And that's important for what I'll show. When we home in on what these thrombi look like, and they can really block the vessel all the way through, this is the portal vein, is that the thrombi attach. You can see how the platelets here in blue go under the surface endothelium and can make contact with parenchymal sites. The thrombi themselves are white, which means platelet rich, but red cell poor. They contain this sort of classical fibrin capricae you'll find from Willebrand fragile, a whole range of different things. And the point here is that they appear at the same time as you see organized, I'm going to call them for simplicity granulomas. They're inflammatory foci, but I'm going to call them granulomas for the purposes of the talk. And here you can see these granulomas are organized structures containing Salmonella in the green dot here, a tiny number of Salmonella for a big granuloma. Macrophages, F480, positive cells, neutrophils written here in red, which are in Ly6G. You can see the inos-rich sites and so on and so forth. And you can basically contain a myriad of different cell types as well. And so this strongly suggested to us when we were looking at this is that the inflammatory response and the thrombosis were tightly linked. And so here we've got vessel occlusion. We can see is that there's, I haven't got 0 to 6, but nothing before here. At day 7, day 21, you see it's long lasting. So for 3 weeks, you've got a persisting thrombosis response in the organ, and it tails off after about a month or so. And those granulomas, you can pretty much superimpose the kinetics on there. And in contrast, if you look at bug numbers, they reach their peak at day 7, which is when you find here, but when you've still got quite a strong thrombosis present, bacterial numbers now are less than 10% or 5% or so of their peak numbers, and then fade afterwards. And it's got nothing to do at all with bacterial numbers in the blood, because these are pretty much undetectable in this model by these later time points. Okay. So other evidence that these were linked. So this was a really good link between the two, but we went and showed a very simple way, is that we infected TLR4 knockout mice, which TLR4 is an immune sensor for LPS, amongst other things. When you infect these mice, they look like non-infected. You don't form granulomas, and then you don't see thrombi in the vessels. And then actually there's a relationship between the TLR4 ligation and the amount of interferon gamma you detect. So it's a really tightly linked relationship here. But in many ways, we need to be careful about what we wish for. And the reason for that is that if we now take a step back and look in other inflammatory mediated models, so here, for instance, we're looking at wild-type mice and interferon gamma knockout mice. Now, these mice have more bacteria at this day 7 time point than wild-type mice. If we look in the tissues, we don't see the granulomas. It pretty much looks like the TLR4 knockout. And if we look in the vessels, in the liver, we pretty much see 0 thrombi in there. So this again delinks further this relationship between the need to have the organism, the inflammatory response, and the level of thrombosis. If we now look in another knockout mouse, and this is a mouse which lacks both TNF receptors on their surface, is that you find is that the bacterial numbers at this day 7 time point are pretty similar, sometimes up, but roughly the same. And then if you look in the tissues a bit more deeply, you get inflammatory foci and accumulation of inflammatory cells. They're obviously a little bit less organized because TNF plays a key role in that. But now you've got same numbers of bugs, inflammatory foci, but no thrombosis. So it tells us is that although inflammation cross-cuts both the development of the granulomas and thrombus formation, that actually you can decouple this in some ways as well. And finally, what if you take the brakes off? So what if you take interleukin 10 away from here? And actually, mostly these mice do better. They have lower bacterial numbers in their tissues at this time. They also make more granulomas. And lastly, they have really, really extensive thrombi all the way through the tissues. And in fact, this is called low dose for a very simple reason, is that we have to lower the dose to be able to not kill these mice with this infection, which is reasonably well tolerated by or survivable by the wild-type mice. So here now, if we take a step back and think about it, if we go all out to control bacterial numbers, that's great, we can easily do that. But think about that nuclear weapon, and then here, there's a price to pay for success. And I'm going to cut to because all this is published. So what we found was that Salmonella infection triggers TLR4, results in interferon gamma. You get an accumulation of monocytic cells, amongst others, protoplanin expression on these, collect T1 platelets, and leading to thrombosis. And we were able to demonstrate this through a whole range of different knockouts and so on and so forth. But there was a problem here, and the problem that was bothering us was that we knew this was a systemic infection. We knew if we looked in sites like the spleen, is that we had roughly the same, maybe even slightly higher bacterial numbers. But at this day 7 time point is that we really would see very, very few thrombi in that organ. So we started to think well, maybe had we missed it because there are so many immune cells in the spleen, maybe we looked at this incorrectly. And Noni was brilliant at really sorting this out. And what she did is that she took a step back and said, well, let's look really early. And so she looked from 8 hours. I'm showing you from 24 hours here, but she looked from 8 hours and so on. And what she found was that actually, if you looked at the spleen in a slightly different way as well, is that at this extremely fast time point, is that the spleen was riddled with thrombi, and you can see this just zoomed in here. They're in the splenic vein here all the way through. Okay. Then tabulate, putting into a figure a huge amount of data. What it shows is that to the single infection, is that the induction of thrombosis was distinct in terms of its kinetics in each individual organ. So in the spleen, you could detect it super quick, but by 24 hours, it would largely resolved and then bounced along the bottom. This probably over-exaggerates what we're seeing. Bounced along the bottom here. But the key point here is that it means it can be extremely quickly, rapidly induced, but also extremely rapidly resolved. And this has implications for stability of thrombus, downstream sequelae, etc., and so on. In contrast, the liver, once it's up, it's largely up. It stays there for weeks afterwards. So there must be at least some distinct features of this response in different organs to the same pathogen. And in terms of sepsis and consequences of sepsis, I think this is incredibly important. So there's predictable and reproducible rounds of thrombosis in a single host to the same pathogen. All right, so if we just, I'm not going to go into this in much detail. All it is that if you look at a thrombus from the spleen and then the liver, they're largely the same. They're platelet rich, they have lots of neutrophils, they have some inflammatory cells, there's plenty of fibrin in there, and then this is just what if you put these all together. Okay. But there's something missing from here, from the story, and that is where is the pathogen? And so it's really a bit player in this story, because one of the things that might have been occurring is that all those bacteria were getting trapped in a thrombus. So this is inside a vessel here where we can see it flanked, and the red here is picking up on platelets, and there's our single organism here. And I love this figure so much. And then, so if we, and Marisol was particularly fantastic at doing this, is that what she did is that she took many thrombi, cut through them each time, looked at them, and did a count for the frequency that you could detect bacteria. And essentially, most of the sections that were cut had 0 bacteria associated inside the thrombus, and about 20% had 1 or more. And then from that, that's about 50% or 60% of these only had 1. So the presence of in fact, that's what that shows. So the bacterial infection was triggering this process, but they were bystanders pretty much thereafter. And so we can look at this in a slightly different way, and this is probably important for what we think is happening. Although you don't find the bacteria within the thrombi, you quite often will find bacteria proximal to the vessels where the thrombi are formed. Now, it's very difficult doing a statistical analysis of this, but it's led us to hypothesize that that's probably the trigger, and this is the crash site, and this is the airbag going off. Okay. And quite nicely, I just like this. This really shows how it's a very thick section of this, with the thrombus here in red, the vessel wall here in blue, and there are 3 bacteria, which if you just look at the vessel, it looks like they're inside, but actually they're on the outside here of this vessel. So if the kinetics of thrombosis are different, then are the mechanisms between the liver and the spleen conserved or different? And so one of the ways that we did this is that we cleared out, we knew from what we'd shown before in the first papers that monocytes were essential for this, or clogenate sensitive cells rather. And then if you knock out clogenate, or if you collagenate treat, wipe out your macrophages, you basically lose all your thrombi formation. So that was conserved. So you need that cell type. If you now break this down to, do you need interferon gamma? And in the liver, it was an essential component. In the spleen, it's probably contributing because of the stats say it is, but it's certainly not essential for this, because you see plenty of thrombi forming and decent sized thrombi forming, irrespective of the presence of interferon gamma. And also, probably really quite surprising to us, and this was this terminal interaction between podoplanin and CLEC2. If you remove CLEC2 from platelets, and then you infect, and then you still see that there's probably a partial effect in the spleen, but it's certainly no more than that. Okay, so that means you've got both a conserved backbone and some variation based on the simply based on the site where the thrombosis is occurring. Okay, so what about neutrophils? So neutrophils, as we can see here, so this is a whole liver section here shown at scale, and you can see the thrombi form within here and stain for neutrophils and fibrinogen, and this sort of like dirty brown color is because the thrombi are extremely rich in both of these factors. Neutrophils are present throughout, and fibrinogen is found within the thrombi as well. Moreover, if you start looking in the spleen or in the liver as to who captures the cells, is that you find that the bacteria are much more likely to be found in a macrophage-like cell or whatever, both in the spleen and in the liver, than you will find them in a neutrophil. And this probably overstates, actually, because this is done by pixel counting, is that when you do it by eye and by hand, you probably find about 3%, 2% of bacteria are found associated with neutrophils, whereas about 95% plus are associated with macrophages. So what this means is that the neutrophils are contributing to this process, but not because they're capturing bacteria. Okay, so what did we do? We do what everyone else does, and we knock them out. And so you can see here, both in the spleen and the liver, and this is published in a recent paper with Marisol's first author, is that if you want the details on how we do this, but in short, if you remove neutrophils, and it doesn't even have to be a complete ablation, is that you of neutrophils, is that you remove the opportunity for thrombi to form. Okay. So now we know the cell types are definitely contributing to this massively, and we can look at this. So this led us then to start really thinking, both in terms of the immunohistology and everything else that we were considering, is how does this really look in the vessels? You know, so we performed intravital imaging of post-infection. So a little bit of a explanation as to what we're looking at here. So platelets here are shown in red. Neutrophils are here in blue, and CD11B here in green. And so this is an infection which has been this is being imaged 6 hours after an infection, and the recording has been made over the period of 1 hour. So what you're going to look at in this video is a 1 hour in this animal's existence. Okay, and you can see here is that although there's a little bit of movement, you've got neutrophils which are already established within the vasculature. You can see here that they're probably at a higher concentration here. There's some interaction occurring with platelets in the vessel. But that actually mostly the platelets still remain free flowing in this at this time point. So you can see this probably thrombus formation starting, but it's not absolutely complete. This other parts here are within the spleen itself, of the red pulp of the spleen. Okay, so this certainly supports the concept that what we're looking at a effect in the vasculature for neutrophils. And if we now take this the next level, and this is a 15-minute recording taken from a mouse which was infected 24 hours before. So this is now when the infection is extremely well established. And now we can see that the thrombi are well-formed platelets move with much slower dynamics and kinetics through the vasculature. That there's again a lot of ongoing interaction between the two different cell types here within the vessel, but to a much lesser degree within the parenchymal sites. Right. So how are neutrophils contributing to this? Well, the obvious way would be through nets, neutrophil extracellular traps, and it is without doubt that we observe the formation and of nets in this response. So they obviously likely to contribute, but if we use a classical approach, which is to add DNAs to the mouse, at least in the experiments that we've done, there's at best a partial effect on this. In some mice respond, some mice don't, but that there's not full activity. So we think probably nets are contributing to this process, but we don't think necessarily that they are the only answer here. Okay. So what could be going on? And I'm only really going to summarize aspects of this data because there's a huge amount of it. It's largely conserved between both sites, but the reasons are probably slightly different. So I'm just going to, and also once you've got the process rolling, is that the downstream effects are probably conserved between, in the basic sense, they're conserved between both sites. So if we take monocytes and neutrophils and infect them in vitro with different levels of bacteria, we find that many of the monocytes that we have in vitro produce TNF, whereas only a marginal proportion or so, it could be assay development, produce TNF after infection. But the big take-home message here is that both cell types can produce TNF. So if you now neutralize TNF, as I showed you before, is that in the liver, is that you've got no thrombosis, but it's also the same in the spleen. So you've got near complete dependence of complete dependence on TNF for this process to occur. So what does TNF do in this context? Well, it looks likely, but if in the same neutralizing experiments, is that TNF is involved in the endothelial activation. This is measured by looking at CD62E distribution along the vasculature. And then furthermore, is that there's also, if you look at the anti-PSGL1 activity, if you endothelial activation can lead to PSGL1. This leads then to, if you neutralize this essentially it helps home cells into the tissues. If you neutralize this, you impair impact the level of thrombosis detected. Interestingly, in nearly every instance, is that we see it's harder to stop thrombosis in the spleen than it is in the liver. Okay, so that's what happens in the absence of any adaptive response in the mouse that or the necessity for any adaptive response in the mouse. And so we're looking at this slightly differently now in human platelets. Okay, do we see something like this with Salmonella mixed with human platelets? And like, if you bang two rocks together, if you mix Salmonella with platelets, washed platelets, whether they're mouse or human, you don't see any activation. It's a fairly clean result that nothing's happening. But you can make this happen, and So this is agrometry, I hate that word, and what it basically means is the amount of, it's a classic way for detecting platelet aggregation, and it basically is about light transmission. And so if you take human platelet-rich plasma and then you mix it with bacteria, what you find is there's a huge spread in the ability of donors to be able to lead to platelet aggregation. It's a huge spectrum altogether. But many can to some degree, and that also links into the lag of this. Oh, sorry, I seem to have reproduced the same thing twice. Okay. So to cut a long story short, is that what we hypothesized and turned out to be correct is that this would correlate to the amount of specific anti-Salmonella antibody that was found within that plasma within those individuals. And then, so what we were able to do that through a range of different ways, I'm only showing one of the ways here, is that if you depleted the specific antibodies from that plasma, is that then you lose the ability to aggregate platelets. Secondly, if you blocked the ability of human IgG to bind to platelets or interact with platelets through Fc gamma receptor 2A, then you also lost the ability to aggregate platelets. So what this led us to conclude is that IgG binding to Salmonella and Fc gamma receptor 2A is required for human platelet activation in vitro. Now, it must be said that the inflammatory cascade type responses I showed you for the mouse are just not possible to be performed in humans in a simple way. So these studies really, really help us in this context. Okay. So, is this relevant for human infections? The short answer is yes. This figure is taken from Andy Pollard's typhoid human challenge model. And in those individuals who got sick, if you followed them and looked at day 14, when we know that thrombosis is still active in the liver, is that these guys have an increase in their in their specific anti-Salmonella IgG responses. I think this is LPS, yeah, so LPS. So what we think is happening here is that Salmonella plus human platelets, if there's no antibody and you don't see aggregation, so there's no basically cross-linking here. If there is antibody present, is that it leads to the aggregation of platelets, and this is entirely dependent upon Fc gamma receptor 2A. It's a big picture type approach, this, is that it says is that it's possible to achieve something here. We're probably missing out on some mechanisms at play. Right, so what does this mean now for in the context of humans? So the first thing is that the thrombi that we observe in the mouse are virtually identical to what we see in the humans. And this is a couple of images here from a study which was published during COVID, and we were shocked. They even have the same shape and the same look, and we confirm this in other ways. So the pathway that we think we've identified for to drive thrombosis in the mouse, we think is highly relevant to what happens in humans. The second way is if we take antibody-mediated effects here, and if we look at COVID in another way, is that individuals with severe COVID are much more likely to have antibodies against this intercellular cement molecule, desmoglein 2, and then individuals who have been hospitalized and in ITU for other reasons, or have had flu, for instance. And in fact, they match patients who have had some presentations of cardiovascular complications here. And then lastly, how does this matter? Is that if we've got cardiac tissue from postmortems from individuals who did not die from COVID or who did die from COVID, and they've been labeled with an anti-GSD2 antibody to pick up desmoglein 2. And basically, this you can see here is that thicker dark bands throughout this tissue. If you look in PM from COVID, this separation, which you can see here, you can see them laddering, is indicative that there's a concern that there has been a separation between these two bands so that the tight junctions are a bit weaker. And lastly is that if you now look in just label for anti-immunoglobulin, so what we're really picking up here is are there anti-DSG2 human antibodies from the host already deposited on this? And then the short answer is yes, you can detect it after COVID. So these aspects of this combine, and hopefully they'll be expanded on in the next step. So just to summarize, it's the complexity and the regulation and all of this relates to its importance. It's to prevent organ and cardiovascular damage is basically the upshot. I'm not going to go through this for reasons of time. But thrombosis develops in different organs with distinct and, incredibly importantly, predictable kinetics to a single pathogen. There's a common mechanistic backbone, but each site has its own organ-specific features. We hypothesize that the trigger is bacteria being proximal to the vessel walls, which zip codes or postcodes, wherever you're from, where thrombi form. We also hypothesize that macrophages play a bigger role in tissues in this process, whereas neutrophils' role is more restricted to the blood. Few bacteria found in there. We've evidence that the human IgG is to O antigen, and this drives human platelet aggregation to Fc gamma receptor. And what we think is that when you have chronic infections, so lots of antigen, at a time when IgG is formed, this may cause thrombosis or at least immunomodulate or modulate in some way. And then bacterial killing, here what we're questioning is whether when we're looking to control infection, how much do we need to focus on bacterial killing versus restricting antigen access to organs? And I'll stop there and thank everyone. I thank the major people. This is obviously a huge team effort. I'm lucky enough just to be the spokesperson, really. Thank you.
Muchas gracias. Thanks a lot. Pueden hacer sus preguntas también en español. La doctora Cristina va a coordinar la sesión de preguntas y ella puede traducir esto. La doctora Cristina Gil, ella es egresada de la Facultad de Química de la UNAM, hizo su maestría y doctorado en el Instituto Politécnico Nacional. Estuvo en estancia en el laboratorio del doctor Adam Cunningham y ahí publicó un artículo muy importante en PNAS, que abrió una brecha muy importante en las investigaciones de vacunas contra la salmonelosis no tifoidea, y después emigró al laboratorio del doctor Burkhard Ludewig, en donde ha desarrollado importantes avances que pronto serán presentados, también parte de ello, y ha ganado varios premios de Syntex y ha publicado en revistas de las más importantes del planeta. So the session is open for questions, guys. Yeah, our students from Egypt. Yeah, you can. The floor is open. And
hi, thank you very much for this great talk, Dr. Cunningham. Very nice to have you here in the National Academy of Medicine. And I know that you are very focused right now about the Salmonella and their effects in this thrombosis related with inflammation, but this is just particular of this kind of bacteria, or we can also expect that other pathogens such as virus or even some parasites could also have this kind of increase endothelial coagulation system overactivation and all that?
Yeah, great question. I think so. So the reason Salmonella is, I use Salmonella for everything, pretty much. The reason it's so important in this context is because you can control the outcome, and by that I mean the survivability of the mouse post-infection. So you can look at it without having to have. You don't need too many bacteria to infect. and you can look over time. So what we think here is that that's exactly what it reflects, is a general principle that can occur. Some of the molecules say, if for Salmonella, you could swap in E. coli, you could swap in Klebs, you could swap in any gram-negative, because obviously they've got a little bit more there, particularly with that TLR4 conserved. Whether you then are able, for gram-positives, they're probably a little bit easier. So there's a structural component here on the bacterial side, is that bacteria spend a lot of their effort trying to not be seen. And for Salmonella, that has a very long O antigen chain. So it means is that to see those, surface molecule, really super proximal surface molecules, just got to try a little bit harder to do that. That's maybe not as the case, for instance, with gram-positives, where you've got a greater dependence on the capsule for to hide from the world, compared to you don't have long O chain LPS. But the principles are certainly the in vitro human stuff, is that the evidence is that many gram-positives are super quick at causing human platelet aggregation in the presence of antibody. So I think that's the case there. The other thing I should point out is that, remember, nominally, all this is happening with Salmonella, which is a nominal intracellular pathogen. So this comes back to, once you kickstart a process, then how much of this is a failure to control, and how much of this is because of exposure to bacterial antigens. There's probably somewhere between between the two, obviously, because no infection, no thrombosis. But you do need, it's really, this is about a particular failure of control to each of these processes. In terms of parasites, again, great question. I suspect, whether we know many infections, parasitic infections, malaria, will have these types of effects. It's extremely well documented, but the reasons why aren't, and that's what I think this brings to the table. Viruses, they're really small, so they're outside of I can't be bothered with them, if I'm perfectly honest, in the main, because they're really hard to model. I find them, COVID in humans suggests that this is a conserved process. Many of the same factors, are present, but there may be more variation for individual viruses.
Thank you, Professor Arriaga. Professor Ludovic?
Yeah, you may call me Burkhardt.
Okay.
We have been friends for so many years. Professor, thank you. Adam, great talk, fantastic. I mean, it is very nicely delineated what appears to be the major conundrum. Why so much fuss if there is only one or sometimes even none of bacteria in the thrombi? Could it be that it's either an overshoot, that there is the initiation still dealing, right? The initial thrombus still dealing with more bacteria, and that that could be one possibility, or you mentioned it before, is that there is a lot of antigen, because you need the antibody, you need the antigen, right? And then, of course, the platelet. So is there the possibility that there's still a lot of antigen around, and that this is then determining also the delay in the overall thrombotic kind of overshooting inflammatory response?
I can deal with the second part first, which is there's one thing I possibly omitted from saying, is that human platelets express Fc gamma, the low affinity, but mouse platelets don't, which is why I was able to say this is in the absence of IgG driving this. But I think there is an antigen. Ironically, we've done a lot of studies, a lot of experiments, staining for bacteria, and we know in some context, ironically, when we don't see thrombosis, is that we can see a huge amount of antigen when the recoverable bacterial numbers, colony-forming units, is extraordinarily low. So that would argue against that this is just simply antigen depot. There may be some minutiae within that the specific antigens aren't picked up by the screening sera that we look at. We know that porins, for instance, are probably if not the first, and certainly in the first couple of antigens which were ever identified to be able to interact with the thrombotic cascade in this way, about 40 years ago. Going back then, if it's an overshoot, again, using sampling beyond just intravital, we've never seen any evidence of that. In videos I didn't show, you can see if you introduce the bacteria at time point 0, they nearly always get mopped up by macrophages. It's just always the case. There's nothing that's happening. The CFU counts in the blood are really low. So we don't really know if it's a pathological consequences. It's conserved across many, as I answered in the last question. The ability to drive a thrombotic response is very heavily conserved across different pathogens. But it looks likely that trade-off between it being beneficial, immunothrombosis versus inflammatory thrombosis, is probably where this comes in, and this may be influenced by dose, localization, etc.
Christina? Thank you.
Thank you very much for the great presentation. I have a question about the kinetics of the generation of the thrombi and also the granuloma and the clearance. Why do you think that these kinetics are so different? Is it something to be, because the bacteria spreads across the organism, so you have bacteria in both the spleen and the liver. So do you think it is something to have with the cells that are sensing the bacteria in that place? You show that the bacteria is mainly kept within macrophages, and the macrophages in the spleen and in the liver are very different. Actually, it has been shown that the macrophages in the liver are like firewall against spreading of LPS. So do you think that is something related with the cells, the kinetics of the bacteria, and why they stay longer in one place than in the other?
I think that most definitely is likely to contribute the type of cells which are present. So we've phenotyped the monocytic lineage cells, if you want to call them that, in the liver in really quite great depth in the original paper. So the upshot is that at time point naught is that you've largely got Kupfers, as you'd expect, maybe a couple of others. But within 24 hours, you've got inflammatory monocytes coming in. By day 7, you've got a really heterogeneous phenotypic presentation of cells, monocytic lineage cells, which are probably a mix of different types. And then how they contribute. So the short answer is I don't know. And thrombosis to anything is common, even lying down, but it's just a very different mechanism. The few surprises is that this wouldn't have been expected to be seen in the venous circulation normally, a white thrombi. So what I think is possibly happening is that the changes in the cell type which are within the tissue, alongside changes in the platelets which are found within tissues, makes a huge amount of difference. If we take the very simple view that is it at a numerical basis, then that's not likely to be important, because when it crashes in the spleen, you've still got cell numbers going up. You're still getting an accumulation of cells in the spleen. So it is something about those. I don't know what that is. In fact, the rate of involution isn't very well explored or understood, particularly in pathological models. What I do think is that maybe taking a step back is that we should be thinking about this in terms of risk. And you've highlighted the bone marrow there. We know that you get a diminution in the numbers of megakaryocytes in the bone marrow immediately after infection. You get a megakaryopoiesis occurring in the spleen, and then it switches into the liver at a later time point during the infection. So it's constantly changing, which may affect the type and quality of platelets that you're producing until it goes back in. But it still doesn't explain why the difference in the spleen and in the liver. So that's probably a really, really long-winded way of me saying, I don't know. But I think the important thing is that by having something to understand the risk and when the risk is that possibly that's, you know, you've probably got different phases of this. Early risk, late risk, when things go back to normal, inverted commas, in the bone marrow.
Thank you very much.
Just what will happen if you have vaccinated animals, like with OMVs or porins, and then you infect them? Will they make the thrombi? What will be the antigens that are triggering these thrombi-related response? Will be LPS or something else?
LPS is the go-to antigen that everyone uses for these types of studies. So LPS can do this, but if you just look at the sheer numbers of LPS molecules which are introduced into a mouse to drive this, it bears no relationship to even the most staggeringly widespread infection that you see. I think, again, you need some organization on the presentation of antigens for this. If you challenge a vaccinated animal, I don't think I've ever seen a thrombus after challenge, which is, again, a really important point, is that vaccination can save lives even through these processes. In fact, flu vaccination drops your risk of cardiovascular events as well. I think the introduction of any antigen probably can drive a thrombosis, but because it's not it's a very short-lived, probably clinically, subclinical presentation of this. But,
yeah, and the role of this marginal zone macrophages, I mean, have you dissect more on the spleen, with which populations of macrophages?
Fantastic question. No, we haven't. We know that they can capture bacteria, so that may be a triggering factor. But so the other thing I didn't show in the interest of time was that you get changes in platelet leukocyte aggregates as well. So platelets are often piggybacking on other cell types, and so you can get changes in there. And to be honest, I can't remember what it showed for the marginal zone, but we haven't been able to break it down to that level of resolution. It's a really important point.
Maybe with the osteopetrotic animals, they lack.
Sorry?
The osteopetrotic mice, they lack the marginal zone, and maybe that could be a model. Yeah, and what you observed, I remember the rosella, the typhi rosella, the rose spots. I mean, that could be one of the mechanisms that we.
Yeah, it could be. I'm not sure if they've ever been dissected in that way, but I can say is that if you look at the intestinal perforation, there's a strong association with thrombosis around that point, and that's associated with the greatest likelihood of poor outcome in typhoid. And you can see that there's. I think the problem is I'm not sure Burkhard had a comment very similar. I think you both did yesterday as well. Is that people say these things and they report, oh yeah, there's a thrombus or there's a heart damage, but getting good quality data to show this at a population scale it's a lot rarer. It just shows what we need.
Excellent, and thank you for this great talk. And Dr. Cristina Hill will